Before we begin looking at the phases of mitosis, there's a little bit of vocabulary that you have to be comfortable with in order to be able to understand mitosis. So one of the things that you need to know is that after the S phase of interphase, DNA is replicated. And when it's replicated, means it has two copies of DNA. So these are two copies of DNA that are attached in the center. That center part is called the centromere. And the two copies of the DNA are called sister chromatids. Now, it is vital to know this because the entire idea behind mitosis is to now separate these two copies of DNA so that one new cell gets one copy and the other new cell gets another copy. So now that we know that, we can look at the stages of mitosis, or the phases of mitosis. The first one is called prophase. Okay, pro is first. Now, there are several things that are happening in this phase. The nucleus starts to disintegrate. Okay, so we did have a nucleus, but now you can see that it is disintegrating. In addition, DNA is normally not condensed. It's not in this kind of X-shaped. Frequently you see chromosomes in an X shape. That is DNA, but that's condensed DNA. And the only time it is condensed is during cell division. So what we've got happening in prophase is DNA that was kind of all like squiggly all over the place, that's chromatin, is condensing to become more like chromosomes. So now we've got chromosomes. We've got different chromosomes. I need more colors. Okay? And they are condensed. They are, remember that when you have an X shape that is made up of two sister chromatids, that's two copies of DNA, that all need to be separated so that one cell gets one copy and the other cell gets another copy. Okay? In addition, you also have centrioles that are starting to release spindle fibers. Now, those spindle fibers are going to attach to the chromosomes, and they're the ones that are orchestrating the entire process of division. They are the ones that are going to pull on sister chromatids so that one chromatid goes to each side of the cell. So let's kind of see what happens after prophase. We move into metaphase. Now, meta, hopefully you know, means middle. So at this point, the nucleus is already completely gone. And what we have is these centrioles, which have attached to our chromosomes and brought them to the center. So they're attached at the centromere. Remember, the centromere is what holds the two sister chromatids together. So they're attached at the centromere. I'm going to fix that in a second. Attached at both sides. Okay. Attached at both sides of the chromosomes. And these spindle fibers are actually pulling on these chromosomes. They're kind of playing tug of war here. They both want to make sure that each side gets one of each of these chromosomes, or one of each of these chromatids. So during anaphase, which is the next phase, notice again that the chromosomes are lined up in the middle of the cell. During anaphase, ana meaning apart, so something is coming apart at this point. Here we have our centrioles. They are pulling apart our chromosomes. So now, instead of having this X shape, we've got one part of the X here, and we've got one part of the X here. Same thing for this one. One part here, and same thing here, one part here, one part here. So those chromosomes have now been separated and they're being pulled towards opposite poles of the cell. Okay, so keep in mind this is a process, and I'm just showing you essentially snapshots throughout the entire process. So once this occurs, we have our final phase of mitosis, which is telophase, in which case you've got the formation of nuclei. Remember that in prophase, our nucleus disintegrated, but now we're making two cells, so we're actually forming two nuclei. Our spindle fibers are pretty much gone. They're done. We've still got our centrioles, but the spindle fibers have done their job. And now each cell has one of each chromosome, or one chromatid from each chromosome, so that what we have now is identical nuclei. Okay, so at this point, karyokinesis or mitosis is complete because we have two separate nuclei, each with one copy of the DNA, of that DNA that was replicated during interphase. Okay, but notice that now we have two nuclei and still only one cell. And now this is not technically part of mitosis because the mitosis is done. We've already got two separate nuclei. But what we do need to still have is cytokinesis. And in cytokinesis, what's going to happen is the cell membrane begins to pinch inward. And so while we have our nuclei here with our DNA, and our DNA does decondense. Remember, it condensed during prophase, so now it has to decondense into chromatin. Okay, so typically DNA does kind of look like a hodgepodge mess like that. And now where you see the cell membrane pinching inward, that is actually, there's a name for that. It is called the cleavage furrow. So if you think about what a cleaver does, it cuts. The cleavage is where the cut is being made, and that is where the two cells are finally splitting. So notice that the cytoplasm would be separating here, and so would any of the organelles that are also in the cell. So here you have kind of an overview of mitosis, going from prophase to metaphase to anaphase to telophase. And then really what happens is cytokinesis starts kind of during telophase or towards the end of telophase. But cytokinesis is its own separate process because it has nothing to do with the nucleus. And keep in mind that mitosis is all about division of the nucleus. Okay. Now, I know that was kind of a lot to take in, but what you do need to do is make sure you are able to go through each of the diagrams, draw your own diagrams, go through them, and describe exactly what is happening, similar to what I have done in this video. Feel free to reference the textbook, feel free to reference any of the videos that are online. There are countless of them, and come to class ready to talk about this process.